Our last speaker in this session, who is from our host institution. His name is Malik Paris, and he's going to talk about confronting the avian flu, the role of synthetic biology.
Thank you, and thanks for inviting me to this meeting. Although I must say that I'm a clinical virologist rather than an exponent of synthetic biology, and the reason I'm here really is that Ralph Barrett twisted my arm so much that it was easier to agree than to refuse. But I just want to look at it from a very practical point of view, how we see synthetic biology or synthetic genes in a much simpler scale than what the previous two speakers talked about in addressing some practical issues in regard to avian flu H5N1. I also want an acknowledgement slide at the end, acknowledging the people who did the work, but right up front, let me say that a lot of this work was done by John Michel Garcia and Isabel Nefkins at the Hong Kong U Pasteur Research Center. So as Ralph pointed out, emerging infectious diseases are with us and will continue to be with us, whether it be AIDS, SARS, antibiotic-resistant bacteria, and the question is, what is next? And certainly one of the problems that does confront us at the moment is the potential of a pandemic, and H5N1 avian flu is certainly a candidate for such a pandemic. So I really want to focus my talk on H5N1, but I think the previous speaker pointed out that one of the viruses that have been completely reconstituted and resuscitated from an extinct state by synthetic biology is the 1918 avian flu virus. I'm not going to talk about that, but I think it would be worth pointing out that that resuscitating this virus has allowed us to understand why that particular virus in 1918 was so virulent for humans, at least in mammalian models, because it is essentially the same H1N1 virus that is circulating in humans now. But the 1918 virus, when you compare it with currently circulating influenza viruses in animal models, has a completely different pathogenesis and pathology and severity. And using these and comparing these allows us to really understand and dissect out the mechanisms of pathogenesis. So let me come back to H5N1 avian flu. As you know, at the moment, at the avian level, in the poultry level, it is entrenched in many countries, over 60 countries, and occasionally it crosses to humans, and at the current point, there are over 380 cases with a fatality rate of 60%. Even though this might be an artificial or skewed increased mortality rate, there is no doubt that this particular virus, in terms of its pathology and virulence for humans, is a very nasty virus, probably even nastier than the 1918 virus. Influenza viruses are single-stranded RNA viruses, the negative sense genome, and in addition to the fact that they are RNA viruses and associated with that, they have a high rate of mutation, just like other RNA viruses like HIV, for example. The genome is in 8 gene segments. The hemagglutinin and the neuraminidase are the proteins on the cell surface, particularly involved in eliciting protective immunity. There are 16 hemagglutinins and 9 neuraminidases, and the one that we are interested in is really the H5N1 virus. The problems that people face when trying to work with a virus like this is that it is highly pathogenic, both for animals and for humans. So it requires biosafety level 3, which restricts the number of people who can work with it. And even if you have these facilities, it certainly reduces very much the amount of things you can do. It is also a select agent being a group of pathogens that are now designated internationally as potential causes of or potential agents for bioterrorism. So therefore, moving any of these viruses from one country to another, or even from one lab to another, is a major operation. It has to go through permits from multiple departments, Department of Trade, as well as Department of Health and takes many months, if at all, to be able to move these viruses. And finally, there are, you might have seen in the media, there are a lot of political pressures now in terms of access to these viruses for study. You might have heard that there are problems of virus sharing, with some countries refusing or being very reluctant to share viruses that are being isolated in their countries for investigation elsewhere. So one of the approaches to try to address some of these issues at least in one respect, is to use what are called pseudoviruses or pseudotypes. So these are essentially viral mimetics which carry a core from another virus, but the surface of the virus, the envelope of the virus, carries the hemagglutinin and the neuraminidase or whatever gene that you want. And this is one of the approaches that we have been trying to take to address some of the issues that we are interested in. So as I said, the principle is that the interior of the virus is an alternative virus, but on the outside, you, for example, can put the H5 hemagglutinin on. There are a number of different backbones that you can use. You can use the HIV backbone, alpha viruses, or a number of others. And there have been a number of envelopes that have been put into these pseudotype viruses. You can also incorporate a reporter gene, for example, luciferase or GFP, for example, in these investigations. So what we have been doing is to create pseudotyped H5 lentiviruses. And essentially, you transfect genes of the HIV backbone together with the H5 hemagglutinin into 293 T cells, and you have to treat this with exogenous neuraminidase because the virus requires neuraminidase to release the virus that is synthesized away from the infected cell. And we have been using luciferase as the reporter gene, and a clade-1 virus from Cambodia as our initial virus for investigation. Now, the problem was when we started trying to do this, or when Isabel started trying to do this, using straightforward cDNA from viral RNA gave very poor efficiency in pseudotyping. However, codon-optimized synthetic genes gave much higher efficiency. So this is why we have been using codon-synthesized synthetic genes for our pseudotyping process. And also, we can develop or we can make viruses only with the hemagglutinin without the neuraminidase. You remember I told you the normal virus particle has the hemagglutinin and the neuraminidase, but for some reasons, for some applications, it is useful to remove the neuraminidase from our virus particle. So that can be done using this approach. This just shows the characterization of the virus particles to show that it contains the hemagglutinin. You can see the virus budding out, these pseudoviruses budding out of infected cells. In the sucrose density gradient fractions, you can see that the luciferase signal correlates with the hemagglutinin and the P24, which is the HIV backbone signal. So essentially, the luciferase signal is being carried by the pseudotype virus. And also looking at to show that these are biologically similar to normal H5N1 viruses. These viruses require sialic acid for entry, so treatment with sialidase, particularly sialidase that removes the alpha-2,3 avian receptor, knocks down the pseudotype virus entry. And the virus entry is also pH dependent, so that ammonium chloride inhibits in a dose-dependent way virus entry. So these are all things that mimic the natural virus infection. And finally, showing that the virus infection is a dose-dependent process. So what we now have is a virus carrying the H5 hemagglutinin. And of course, the viral replication process involves many steps. It involves attachment, it involves internalization, and then the viral replication process and the viral egress process. So clearly, we are not trying to address these parts of the viral replication cycle, which are pseudotype. We are only addressing questions pertaining to this early attachment and entry process. So we have the pseudotype, we can infect MDCK cells, and you can detect that infection with a reporter gene. And we can use these particles to address questions at the early attachment stage, to address questions of viral receptor. You can also develop methods for detecting antibody. This is for diagnostic purposes, antibody in patient serum, because if you neutralize this pseudotyped virus, you will basically block the reporter gene signal, so you have virus neutralization. And also these pseudotypes can be used for high-throughput antiviral drug screens. So these really are the three things that we have been addressing, and what I will briefly tell you in the next few minutes. So firstly, antibody tests, serodiagnosis. The standard way of doing it is to do this with live virus, to do neutralization of live virus. But as I said, this is a BSL-3 requiring process, and it's quite a tedious process. But here, what we are showing is that these viral pseudotypes behave perfectly in these viral neutralization assays. So these are over 100 sera from control people who should not have antibodies to H5N1, and most of them do not. A few of them, just 2 or 3 of them, you can see do. And in fact, these patients or these people actually also have antibodies by the neutralization test. So this is not really false positivity. It is really that a few people, particularly the older people, do have antibodies to H5N1, or at least H5 in this case. On the other hand, if you take sera from patients with H5N1 disease, all of them efficiently neutralize the pseudotype. And we have optimized this in terms of the optimal dose that is required to, because if you have a lower input of the pseudotyped virus, you have higher antibody levels. But once you increase the dose up to a certain point, then the antibody titer becomes independent of viral input. And these are sera from a series of patients from Vietnam with H5N1 disease, and you can see the individual serum samples and a fitted curve, and the blue line is the neutralization conventional test, and the red line is the pseudotype assay, and you can see the two are very well correlated. And again, here you can see the correlation between the standard microneutralization test and the pseudotype assay. You can see it's slightly more sensitive than the microneutralization test. And the reproducibility and diagnostic parameters are quite satisfactory. So we certainly propose this as something that can be used for a diagnostic purpose in a BSL-2 environment. It's a functional assay. It's biologically active in looking at neutralizing the virus. It can be used in a high throughput fashion. And the most important thing, I think, is because you can essentially make this with a synthetic gene, you do not need access to the virus, nor even to viral RNA to produce these pseudoparticles. So they can be basically made as long as viral sequence data is out there in the gene bank. And also, as I said, you can dissect out the antibody responses to the H5 hemagglutinin from the antibody responses, if any, to the N1 neuraminidase. The second application that we have for this is to look at high-throughput drug screening. And essentially, now we are using this pseudotype virus with the luciferase reporter gene and using a range of drugs from drug libraries to look for active compounds that will block virus infection. And this is ongoing work, but just to point out that this certainly has been possible. So my colleague John Michel Garcia has screened over 48,000 compounds in a cocktail fashion, and has got a number of hits that interrupt virus entry and infection. And while these are still being worked up, I think it is important to note that the currently available antivirals for influenza act at this stage of the virus cycle, that is the uncoating of the virus, or in the release of the virus. So the uncoating of the virus is the amantadines. The release of the virus is the oseltamivir, the Tamiflu that you have heard about. Whereas these hits that we have got, by definition, are working at this phase. So in other words, they are different to the currently established antiviral drugs. So finally, I want to address the issue of the virus-receptor interaction, which again, these pseudoviruses or virus-like particles allow us to do. At the moment, you have this avian virus, H5N1, infecting avian species. Human viruses, the seasonal influenza viruses that we have, infect us. And it's very rarely that an avian virus infects a human or that a human virus infects an avian. It does happen very occasionally with H5N1 with disastrous consequences, but there is a very strong species barrier that separates avian influenza viruses from infecting humans. So the question is, what is this species barrier due to? And clearly this is a polygenic trait involving a number of genes of the virus, but certainly the receptor is certainly thought to be an important component of this barrier. As Ralph showed you earlier, in the case of SARS, the receptor seems to be a very dominant factor that affects interspecies transmission. So the paradigm that has been established for flu is that there are sialic acids, which are the receptors for both avian and human influenza viruses, but the alpha-2,3 linked sialic acids recognize avian influenza viruses. The alpha-2,6 linked sialic acids recognize human influenza viruses. And it was believed that humans predominantly have the alpha-2,6 receptor, whereas avians have the alpha-2,3 receptor. So because of this receptor difference, the H5N1 avian virus finds it difficult to infect humans. Although it is known that the human lung, lower down in the lung, you do have this avian receptor present as well. So if you use conventional methods, which is basically lectin binding to differentiate these different sialic acids, and you look in the upper respiratory tract, you really don't see evidence of this avian receptor. So this is actually using one of these lectins that selectively recognize this alpha-2,3 receptor, essentially totally clean. There's no evidence of this detectable in the upper respiratory tract. However, if you actually take biopsies of the human upper respiratory tract and infect them, if you infect them with human flu, they infect nicely as you would expect. But surprisingly, if you infect them with avian flu, H5N1, they equally infect. So even though the so-called receptor is not present, the virus does seem to infect. So the question then is, how does this H5N1 virus infect the upper respiratory tract in the absence of the conventional established receptor for this virus? Are there alternative receptors? So this raises questions of what really are the glycans that H5N1 viruses bind to, and what are the genetic mutations in the viral hemagglutinin that affect these receptor binding for these different glycans, and how can we investigate this safely without doing some of these things in a BSL-3 setting? Because some of the approaches that one would like to take, really one cannot do within a BSL-3 setting. One of the approaches that has been taken by others recently is to use these glycan arrays. So rather than just look at these two alpha-2,3 and alpha-2,6 glycans or sialic acids that I told you about, they have spotted a whole range of glycans. And you can see, for example, down here, this is a human virus, and this green bar shaded section are the so-called alpha-2,3 receptors, and the yellow shaded section is the avian type receptors. And you can see the human virus recognizes a number of glycans in this human region, whereas a typical avian virus does not recognize any of these glycans. However, if you take H5N1 on these glycan arrays, you can see that although it generally holds true, certainly H5N1 does recognize some of these glycans that fall within this green area. So we were interested really to try to address this in a more detailed way. And one of the approaches that one can use is to use saturation transfer difference NMR. The principle is that you irradiate the virus, if you like, or the virus particle, and you mix them with the ligands, sialic acids of different conformations that you want to put in there, that you are trying to find out which ligand is binding to the hemagglutinin. And the ligand that does bind, the energy will be transferred to this, and it's a reversible reaction, and you can then actually see the ligand that has interacted with the particle that has been irradiated. Now, of course, you can't do this with the live H5N1 virus because it's too dangerous to handle in an open setting like this. But you can do this with virus-like particles, H5 virus-like particles. And this is exactly what we have done. And we have essentially shown proof of concept that this system works. And in addition to showing that, for example, one particular sialic acid rather than another one binds to the virus-like particle, you can actually map very precisely the molecular contacts of the glycan and the hemagglutinin. So this is just to show more detailed analysis of optimization of this process. So in the early studies, there were a lot of impurities that gave us a lot of false signals, but now we have quite a clean signal in this system. So using these synthetic H5 VLPs, we can interrogate the interaction of a range of glycans found in the human respiratory tract, and also investigate the mutations of H5 hemagglutinin on receptor binding. Just to end by acknowledging many of the people, as I say, who did the work, John Michel Garcia and Isabel Nefkins, Ralph Altmaier, and others at the Hong Kong U Pasteur Center. The STD-NMR work was done in collaboration with Mark von Itzstein at Griffith University. And in the clinical aspects for human sera with H5N1 patients, we had collaboration from Meno de Jong in Vietnam, Philippe Bouchy in Cambodia, and vaccinated sera from Sanofi Pasteur. Thank you.
Okay, thank you very much, Malik. So I want to make sure that everyone can catch the bus, but I think we have time for just one or two questions. Ralph.
Sialic acid can be modified a variety of different ways. Have you looked to see if different H5 strains from different years bind different modified versions of either the avian or the human sialic acid moieties?
Yes. So you can look at it from two directions. You can look at different glycans to see which of them bind to the H5 virus. You can also look at mutations in the H5 virus and to see how that impacts on sialic acid recognition. Now, over time, there really has not been a change in the consensus, if you like, of H5 hemagglutinin in recognizing more this alpha-2,3 sialic acid. But within an individual person, if you look at the quasi-species, there certainly does seem to be minority populations that have mutations that do recognize the human sialic acid moiety better. So that probably makes sense. I mean, the virus is trying to establish within the human host, and it's trying to switch its receptor binding profile. But I think what I was trying to get at is that we believe that the dogma per se needs to be re-looked at, because the upper respiratory tract, as I say, does not have this so-called conventional receptor. So it is a glycan. It's probably a sialic acid, but it's probably something else. So that's one of the reasons why we think the argument has to be looked at afresh, and that's our interest in this area.
Any other questions? Okay, why don't you join me in thanking all the speakers for a really fascinating session.